My name is Marla Phillips. I'm director at Xavier Health at Xavier University, and we're very pleased to have you join us for this free webinar. I would like to thank our sponsor. Both Troy and Melissa, our speakers, are from Compliance Insight, and they are sponsoring our ability to do this. So it's free so that you can join it, but at the same time, we do need some financial resources at our little university so that we can exist. So we do appreciate the financial support to be able to bring this great content to you. On the next slide, I would like to introduce our speakers so then they can take it from here and you don't have to listen to me anymore, right? So Troy Fugate is a vice president at Compliance Insight, and I believe a co-founder. Isn't it right, Troy?
That's correct.
Yeah, see, I knew that. All right. So Troy has over 30 years of experience and in the industry. He started off, we were just talking as we were getting ready to start today. He helped FDA do investigations back during the generic drug scandal. So he was doing some criminal investigations back then with them, but also has tremendous experience from companies like Watson and Pfizer and Perrigo and Roche, and just brings tremendous insight. And most recently has been working with companies in the cell gene therapy space. Melissa Schneider is a manager of regulatory affairs at Compliance Insight. She also has 30 years of experience, most recently coming from the Cincinnati Children's Hospital Medical Center, where she was doing work in cell gene therapy. And by the way, the Cincinnati Children's Hospital is one of the top 3 pediatric hospitals in the nation. So both of these speakers bring tremendous experience and insight that we're all looking forward to hearing. So at this point, I'm going to turn it over to you.
All right, great. Thank you. You know, I have to say, when you said 30 years experience, that really sounded really old. So,
well, it is, but I'm right there with you. So what can you say, right?
Yeah. So thank you for this. And let me just jump right into this. I can say the thing that Melissa and I were discussing with this is just the phase appropriate implementation of GMPs for this industry that's blooming like crazy, and it's always a question that we're getting, and even just this week I've had 3 or 4 questions about what do we do, how do we do it, and it really comes down to a few points with this. It's like a pendulum. I always put it in a pendulum, sort of in your mindset. It's too much, too little. Where does it go? Is it uniform? Do you have risk controls in place? Do you have risk philosophies in place? How do you start planning? Where do you start planning? What are the options for this? So we thought, let's go in and put this together and start talking to people about it.
All right, so the next couple of slides, we've actually taken a look at the list of who is going to attend today, and really kind of broken it into 2 categories. So we've got a lot of people who are actually in the cell and gene therapy industry and have quite a bit of experience in that industry. So really understand the processes of cell and gene therapy. And then Troy, if you'd advance, then we have the group who's in the pharmaceutical industry. So part of our presentation is going to be discussing CGMPs and the phasing in of CGMPs into our cell and gene therapy processes. So the pharmaceutical industry people are really going to understand more about the CGMPs and CGMP implementation. So these are really our 2 categories of attendees. And so for those pharmaceutical industry people, I wanted to go through some very basics of what is gene therapy, what is cell therapy, and how are those therapies administered. So this first slide, what is gene therapy? So the basic definition is really manipulation of genetic material. And when we're talking about genetic material, specifically, we're talking about DNA or RNA. And when we're manipulating it, we're talking about introducing it or removing it or changing it. And these are the DNA or the RNA in the cells of the patient. And when we're manipulating it or introducing it, removing it, or changing it, it's to treat a disease. whether that disease be an inherited disease or a developed disease. So this diagram I have here is an example of the CAR T process. So it really starts on the far left of the slide with apheresis. And it's pulling the patient's blood cells from the patient during the apheresis process. And then we move on to this first portion of the process. Our little orange and blue circles there would be the patient cells or blood cells, and it goes through a sorting process. So in the CAR T process, we would be sorting out the specific CAR T cells, which are our little blue circles there. We move those cells into the lab, and they would go through an engineering process, a laboratory process, to actually do that manipulation of the genetic material and change those genes. They would go through an expansion phase in the lab, so the growing of the cells to expand the number of CAR T cells, because we want to have a specific number of cells before we put that material back into the patient. And then on the far right, the infusion, the cells go back into the patient. All right, Troy, next slide. All right, so another route of administration, another commonly used method is a vector. So in the last slide, we talked about manipulation of RNA or DNA. This can be done by using a vector, and a vector can be delivered directly into the body. It's commonly a virus, which can be delivered into the body, or the vector can be used in the cell therapy. So cell therapy is using cells from the patient, which would be autologous use, or cells from a donor, which would be allogeneic use. And those cells are then modified and then put back into the body. So as I discussed on the previous slide, in cell therapy, those cells are manipulated or changed before being infused back into the patient. All right, Troy, next slide. So lastly, our third type of gene therapy administration, we have gene editing. So gene editing is a highly precise technology. It removes or corrects pieces of the DNA rather than replacing the gene. So in traditional gene therapy, we're actually replacing the gene in the body. With gene editing, we're correcting the gene. So this can be done within the patient body or outside of the patient body. And again, it can be done with the patient cells or with donor cells. And a common method is what I have listed here, CRISPR. So with that, that gives you a little bit of background on the process of gene therapy, or I should say processes, since we have multiple processes. And I'm going to turn it back over to Troy, and he's going to start talking to you a little bit about the CGMPs and phasing those in.
Okay, excellent. Thank you. Thank you. Before I get into that, just so people are aware of the magnitude of the market, the people are out there working, companies coming up with this, a lot of it in academia. If you look at where it started back 2016, where this chart started, just the different phases, and where we are now in 2021, how significantly it has grown. And definitely I see this out here as to what is in the pipeline coming up. And if you notice it going out to the next almost 10 years, it increases significantly. So there's a lot of people out there starting new. They're starting to put things together. They're starting these plans. I put this here just because it is so imperative now to start thinking about GMP implementation. And I use the term GMPs just from the standpoint of concepts of control. I'll talk about that as we go through this, but our opening statement with this, all right, we're giving you our views of what's needed. This is an introduction. This is a lunch and learn type of approach. You have to take this and put it into your company's system, into your unique environment and the context and culture of your organization, your product or products, your company, however the vision is of that. Make sure you fit this into your organization. Really what we're doing here is igniting a thought. We're planting a seed in your mind to say, let's start thinking about this. How do we do this? How do we do it better? So with that in mind, let's take Covey's approach, Stephen Covey's approach in the 7 habits of beginning with the end in mind. So without that, we just go toward a goal that we may not really understand what it is. So I'm asking you, before you start with all this stuff, before you start with anything really, is understand, define success. What does it look like? Where are we going with this process? And I put some points here, just some thoughts. What is the goal of the product? What are we going to do with that? Where are we going with this? Is it just to develop it, hand it off, sell it? Is it to take it to market? What is it? How do we do these things? What's the company's goal out of this? What is their mission? What is their vision? And what level of risk are we willing to accept on these things? Trust me out here, I've seen a lot of companies come in various levels of people, very smart people, and they're trying to implement too much, too early, too little, too late. And so you've got to understand what is the risk that we're willing to accept from development all the way through clinical, through the validation, and then into the commercialization. So start thinking about that. And Melissa and I spoke about this at length. This is our best advice to you. If you get anything out of this, it is this one item. Have a policy or procedure which outlines your approach. If you don't have one, put something together. Start thinking on it. Start putting something together. Document your rationale for why you do this. Just the concepts, the philosophy with it. If you don't, then it's just sort of you're doing things and it causes some issues. And I'll talk about that here in a minute. But you want to make sure you document your rationale. For one, it keeps everybody marching into the same drum beat with this thing. So let's get started with this, the meat of this thing. Okay. What do you need to start thinking about? The scope of what am I trying to control with GMP implementation? This is just sort of starting the list of things to start thinking of you got to get them into control. The raw materials. How do you get them? What do you do with them? What's the grade? Where does it change as I go along? How do I know they're in control? The components, the same philosophy with that. The methods, test methods, in process, finished product, the processes. How do I put these together for drug substance to drug product? How do I think about these things? Do I have contract testing labs? Do I have contract manufacturing organizations? A lot of companies out there are smaller. They're going out to CMOs or CDMOs to have them make the product. And sometimes we just want you to do it. Sometimes we want you to start it and then we'll take it over once we get our facility built. All sorts of things out there being worked on. Contract quality organization, which Melissa and I happen to be part of one, coming in and helping you understand where is the level, what are the regulatory implications, dealing with the FDA, the validation departments, quality departments, all those departments have to be in the scope of this thing. Definitely research and development, definitely tech services, tech transfer. documentation of all of these things. And lastly, I put down there data integrity. Make sure you have data integrity. That is a massive thing with the agency. Looking at this is making sure what you put together from beginning to whatever endpoint you have, the integrity of that data is maintained and controlled. Think about Alcoa as you go into this thing. So the graph of the concept of this, right? So I put it together in a DNA, Melissa and I were looking at it, it's like, all right, let's put it together as a DNA strand. So the concept that we're talking about here is to increase the quality system implementation as you progress along this timeline from left to right, is starting to wherever the endpoint is. And this one I put together as commercialization. You'll see I map it out here as we go along. This requires you to understand the goals of the company. Where is it that we want to go with this? And you map out then your process along this timeline. So let's look at what typically is done, right? You start with some type of R&D. You think about things, you put stuff together. Sometimes people go to tox, preclinical studies, phase 1, 2, you go to phase 3, sometimes 4. Then you go to PPQ, which is your process validation. And then you go to commercialization, or you go to market. And then there's going to be an FDA approval process down toward the end. All along this way, you should definitely have some FDA involvement. You should certainly understand what it is that's required for each of those things. So let's go on with this. Where we go wrong, always the pendulum, right? We apply too much GMP too early in the process. You're trying to control to a commercialization process, something that you're working on up front. It's too much. You can't even move. It creates problems there. We wait until we get too late into the process to start thinking about GMPs. We wait until we get down to the wire and we say, all right, I've got to start thinking about GMPs. At that point, you're going to probably uncover a multitude of issues. And we're inconsistent between products or processes. I can tell you, you can have a company, multiple products going on, and you have different levels of compliance, different levels of implementation along. Each of those things are done by different teams, and they have different philosophies. That gets you into trouble. So, biggest items out of all of this, it confuses people, especially people looking at one team that's advancing and another team that's not. And they're looking at each other and they're like, how can you do that? We can't. You can't make product. You can't make things happen. You're stuck. You're in a too much of a grind. It becomes a catch-22. You're rushing at the end to wrap up the GMP issues. You're trying to justify things. You're trying to say, these are all things that are okay. We just didn't do them up front. We learned our lesson and we won't do it again. Promise, promise, we'll do that. You get filing delays. It causes concerns for product quality, patient safety as you go through this stuff, especially as you get into some of the clinical phases where you're dealing with people, and regulatory inspection issues as to how did you develop it? Why did you do certain things? How did you keep it under control? Those cause questions, especially as you're filing, maybe a rolling filing with it, or you're talking to the agency and they have questions about data and you're not able to explain it. These can all be very problematic. So let's get started with this thing. We start with R&D. Let me start out with this, okay? R&D does not mean zero control, right? Does not mean zero GMPs. You have to have something in place. I put some points down here, the things that I typically want to see. And Melissa, you certainly were involved with a lot of the work at Children's, but the thing that we were talking about here is GDP. Document your stuff. I'm not talking about making sure every T is crossed and I is dotted, but it's document why you did certain things. What's your plan? What did you find when you did these things? How did you make a decision to advance or to do something else? You did testing in one thing, you found out that really wasn't meaningful, so you dropped it. Document why. Just put it down. Doesn't have to be war and peace. It can be very simple, but document it, document it, document it. Good supply practices in place. At this point in the timeline, early on, just make sure you get things that can be controlled later on. What you don't want to do is buy something in the R&D phase. You get media from Troy's Media and Barn Door Company, right? I sell you media. It's cheap. That's what you're looking for. You get in, you say, oh, this really works well. You come back later on and you say, well, I want to take this over to clinical phases, and we're going to end up taking this into commercialization. And I say, are you kidding me? I make this, I'm a Media and Barn Door Company. I make this in a shed. I can't do that. So now you're problematic with it. So make sure you can control these things. And I'm going to pitch this over to Marla. There is a good supply practice program that was put together years ago. We worked on it that would certainly help out with this. So if you don't have that, reach out and look into something like that. And lastly with this, talk about the equipment with this. You want to make sure things work right. Too often I've seen R&D as sort of the stepchild of equipment, processes, material. You get expired material. Go play with this stuff. We had a whole bunch of it. It all expired. You go work on it. We don't calibrate the equipment. We don't have the best equipment. We don't have the best facilities. And so you develop these processes with things that are very problematic to repeat because you may have been using equipment, you may have been using material that wasn't really right. So think about what it is you have to control here and then control it. Doesn't mean you can't change it later on as you may come with finding experience, but it does mean that you put some effort and thought into it. So as we progress down the line with this thing, we're now going from thinking about it, playing with stuff. Now we're like, well, we got something we want to think on. We want to take into place with this thing. You know, what do we do at this point with the tox things? And a lot of this stuff is you're using contract labs at this point. Make sure they understand what it is you want. What are you looking for? What are you trying to do? Be very specific. Work with them, especially if they worked with other companies. Have them help you. What should we be looking at? What are we trying to do? Again, it's that mission philosophy. What am I trying to do with this part of it? Be very upfront with them and put a protocol together. Put something together. It's simple. It's saying what you're trying to do, what it is you're looking for, when are you going to get the data? What do you do with the data? How do you react with it? At this phase, you know, we could start thinking about test methods, some of the thinking on it. What are we doing with this thing? How do we know we're in control? How do we test these things? And I go back to my early career with this, with the generic drug scandal and the Barr decision. Judge Wolin in the Barr decision said that testing itself is the heart of compliant pharmaceutical operations. So if you take that into consideration, this is the beginning of the heart of this entire operation. How do I know I'm in control? I've got to have accurate tests that give me the data. Otherwise, as a quality person, I don't know. I don't know what it means if I'm looking at data as variable and it's all over the place. So always start thinking about it at this stage of the game. All right. What do I have to do? Do I have to qualify methods? Do I have to put things together? Whatever it is, I'm not saying you have to do it at this phase. You have to start thinking about it back through here. I want to make sure you get data that you can make true decisions on based on the data. So go on with this qualification and validation. And I bring this up because I was talking about this back here. So the qualification and the validation aspect of it is different, all right? Qualification It's proving and documenting equipment work, right? It's installed, it works correctly. You get some of the data out of it that you can look at and you say it's good. Validation is a bit higher step with this thing. This is the same philosophy of putting in GMP along a gradient type of approach. So you qualify things and you validate things. There is definitely some guidance documents out there, the ICH, USP, FDA docs. If you think of all these things, what is qualification, what is validation, as you go through these phases. And so clinical validation can demonstrate the effectiveness of the test. And understand what I'm saying here, the effectiveness of the test. How relevant is the test measurement to clinical conditions? Don't know. What does it mean? Again, I'm wanting you to think, what is it I'm trying to do? What am I doing at this phase? How do I know I'm going to be in control? How do I know I'm going to be successful? This is where we're planting these seeds to make you start thinking about this. So progressing down the line with this thing, we go from now with R&D to the tox, preclinical type of studies, safety studies. Now we're getting into phase 1. Now you start thinking about, okay, how do I start controlling the process at this stage? And I'm not saying you lock it down. I'm saying, how do I start thinking about it? What really makes things tick with this? You're starting to think about robustness of the process. How do I control it? Where are the control points? If you don't, you start considering evolving the process at this phase into something else that, again, is where am I taking this? If I'm going to take this down to market and to the PPQ process, I'm going to have it locked down and in control at that phase. So if I start thinking about it here, it gives me time to start dialing things in, understanding it, putting some pressure on things to say, I want to control it. By the time I get to this point, I want it locked in. So if you're still at this phase talking about FIO, for information only testing, that's fine, but you need to start thinking about what I do with that data. One thing that will definitely get you into some serious issues with this is carrying FIO testing all the way through and you get down in some of the later phases of clinical operation. The thing that I constantly ask, I come in.
Yeah, just a share.
Whoop, someone have a question? Nope. Okay. So FIO testing, I get there, I look at it, I say, what are you doing with the data? And I tell you, about 75% of it is where we're collecting it to see if there's anything that's going on there. And I say, okay, great. Where's the protocol? What are you going to do with this information? Who's collecting it? Who's reviewing it? How is it being reviewed? And typically I get a blank stare. Be very careful with this because I constantly go through looking at companies and I see these FIO testing and I say, show me the data. They bring me the data. I look at it and I see it's all over the place or it hasn't been tested. There are issues with the data. It's one extreme to the other. I say, what does this mean? And people don't know. Have a protocol in place. What are you going to do with the data? How long do you collect the data? Where do you make a decision? And this is good, we're going to continue the test, or we're going to drop it. You got to have something in place with that. And I say that again and again because that's one that keeps coming back to haunt people. So underscore that one. If you're still gathering data and what makes the product tick in this point, it's great. That's what you're there for. You're looking at this. Again, this is you're starting to understand the process. You're dialing some things in. Think about this, okay? Cautionary with this. If you're performing a bench scale, lab process at this point, and you don't start to change it now, and you don't start thinking about how to change it now, it becomes increasingly more difficult and problematic and time sensitive to change it the farther down you go on this timeline. Start thinking about it now. A lot of times people are coming into this and it's very put together in pieces from a lab process, an R&D process, which is fine, but you have to start thinking, how do I make this work in a commercial operation or something along that line where all of these aspects are controlled? Start thinking about it now. All right. What do you need for phase one? All right. What's the test method? Describe it. Tell me about it. How did you select it? You may, and remember here, you may have selected a wrong method. You may look at it and say, hey, this really doesn't tell us what we think. We have to go with something else. That's fine. Detail it. Put it in your change control process. Talk about why you did it. Put the data there. Have this stuff in place so that you can truly say, yeah, I made this change with reason, with intent. And here's the data saying why. Test methods are designed to show all these things are listed here, right? Product does what you think it does. The process is consistent. Again, we're starting to think about philosophies of endpoint here, taking it to market. We're starting to think about impurities. We're starting to think about sterility. Very critical at this point. You're starting to think about these things we talk about here, and it's highlighted: safe, pure, and effective. You got to make sure you start thinking about this stuff now with the SQIP concept, right? These are the things that the GMP look at: safe, quality, identity, purity, potency. These are all things that have to come about. And now, again, you think about this, where we are with this, and think about it now. It'll be much better later on. So somewhere in this chain, typically around back through here, and it depends where you are, how much you want to do with this, but you can start having engineering runs. You can start having shakedown runs. You can start having trial runs, whatever it is that comes up. Think about what it is you're trying to do. What is the appropriate level of GMP for these types of runs? And again, make sure they're phase appropriate. Make sure they're appropriate for what you're trying to do, the intent of that particular operation. What are you trying to do? What's needed to control it, to get an answer out of it? There has to be some level of control at this point. You have to look at it from that perspective. It's not just Wild West here. Go out, run it, and do it, and we'll get some data back. And hopefully it'll be good. You got to have some level there, just from the basic concepts, training of those things, documentation of this stuff, the material. Is it released to the level we need it? Not maybe full testing, maybe just C of A type of release. You want to make sure you control it to the level you have to in these types of runs. And bear in mind, all right, some of this data goes into the filing. So what's that appropriate level of GMP you need for that? If I have stability data that I gather from this, where do I need with that process? I need to document, I need to control this stuff. I need to review records. I need to have that control in place. Where is a decision point for some of these things? It may be pivotal batches type of things. A decision point has been made on how to proceed with this. Think about GMP controls in this place as warranted to meet your goal. I often say with this is typically not a human use lot back at this point. When you're running these things, this is typically not that level. I can't say it's always 100% that level, right? Two things 100%, death and taxes. This is something you have to start thinking on. So let's go on down the line with this thing. We'll go on from phase one, going down the line with this. You keep improving the levels of control at this level. You're going through, you're saying, all right, now I have to have certain things I want to make sure I understand. Quality attributes, critical quality attributes. How does the product work? I'm starting to dial it in. I'm getting ready now for definitely human use. I'm definitely starting to think about in game with this, of validating the processes. Learn from your experiences with this. Learn from your experiences means you've documented your experiences, because people leave companies. If you're relying on a person that knows the process and they're the ones that know everything, and they win the lottery and leave, or they're swept away by another company, what are you going to do? Document this stuff. Learn from your experiences. More control over the raw materials at this phase. We're starting to now think about sterility of testing of materials coming in, maybe particulate, maybe functionality of that material. Maybe there's something unique about your process that you have to test above and beyond what the manufacturer tests. So make sure you keep that stuff in mind. Do you have a raw material program? Maybe you don't. Maybe you need to start thinking about it at this phase, maybe even earlier. Do you audit these people? Do you visit them? Do you have service agreements in place? Have to make sure that you think along these things. Again, the changes that you do with this stuff, you have to document this stuff. Why you do it. And I tell you that again. Why did you do it? Not just because. There's got to be rationale. Document it, put it together. Again, it doesn't have to be giant reams of data. It just has to have, here's the data to support it. This is why we're doing it. Document that so you have this type of thing. Do you have to inform regulatory agencies of updates? Maybe you've got a rolling filing. Maybe you've got something going on. Maybe regulatory at this point is getting the filing ready. Make sure you start involving them at this phase, right? Raw materials, I spoke about that earlier. Make sure you start thinking about the C of A that comes in. Maybe start testing some things. Maybe you test a full regimen. I don't know. Think about what it is you want to do. When you get typically down to phase 3, you typically are having incoming testing of some degree on components and raw materials. So let's keep going down. By the time you're getting down to this phase of the line, you're looking at some validation of the process, methodology. You're wanting to start thinking about what are the things I need to put together in a validation setting, which means you get the validation group involved. You start thinking about what are critical to quality? What are the quality attributes I have to start thinking on? Where do I start measuring things and why? Methods, how do I have those in place? Are they qualified? Is qualification sufficient? Do I have to validate them? Do I have to qualify and validate equipment? All those types of philosophies. If you do everything right, by the time you start getting down to this phase of the line, all right, you're going to be talking about FDA discussions, really pointed ones now of I'm validating the process. Look at the things here, FDA discussions, control to the appropriate level of understanding of increasing control as we go along this thing. Make sure you've got that down. Getting the BLA ready, I spoke about it, making sure the regulatory group is involved with this. What are you trying to do? How do you do it? What are the meetings with the agency so far? What is it that you should be doing to get ready? That final point, the FDA approval for market. And dealing with this stuff as we go along. There will always be issues. Maybe we put failure here. You know, I often say it's an opportunity to understand more about the uniqueness of your product or your process. So what does it look like, right? In this type of environment, you could have things that are very fast processed through. So you may find out that something didn't work as well or at all. How do you measure that? You might have patients at this point, especially in what a lot of people are dealing with, very critical diseases, that the patient passes away. What do you, was it something that we did, something that caused a problem? You got to start thinking about that. How is this really impactful? Do you have data to say, no, this lot went to other patients, no issues with them. We've tested it. Everything's great. We didn't cause an issue with this. What about out of specification testing, post-administration? What do you do when you look at something like that? If you've gone through, this is the old classic pharma world of radiopharmaceuticals, right? Radioactive. So you process this stuff through. It's on an airplane being dosed to the patient before the testing is done. So that is something that's out there for you to look at in this industry, to go through and say, is there something we can learn from lessons from the past? I can tell you, hard learned lessons from the past. What do we do with this type of thing? How do you investigate a failure? Think about it. What do you do? How do you put this stuff together? And then again, what does post-market surveillance look like? Patient monitoring. What do you do when there's an issue? How do we just know the patient is doing well? Melissa brought up the point, the FDA, and you were dealing with this, requiring 15 years of patient follow-up. Think about that. 15 years. How do you collect the data? Because where we are right now, 15 years from now, dealing with the technology we have, it's going to be antiquated. How do you store it? Where do you get this data? How can you retrieve the data? And 15 years ago, I can tell you, I was storing things on floppy drives, things that now a thumb drive. I don't think 15 years from now, there probably will be a thumb drive. And so you've got to be able to collect that data and put it together. So helpful hints with this, right? Going along the journey, and this is truly a journey. Helpful hints with it. We put this together, and I thank Melissa for this. Have a policy. What does it look like? Use the ICH Q7A as a guide. There's a table in that guide. It talks about, and this is API guide for pharmaceuticals, that the gradient of GMP implementation as you go across something that may come from a field, from an animal, from the earth. You can't have full GMPs from a person in a field collecting material. You've got to have that gradient, that philosophy of how do I implement an ever-increasing level of GMPs to a final point. Some things paramount to have with this, all right? Number one, data integrity. Number two, sterility for patients. If you go into people, make sure you don't have a problem with that. And let's just call it generally here safety. And then keeping good records. Without that last point, you're going to come up with something that is problematic. Even I'm talking to other people about getting to a phase 2 and then tech transfer it out and sell it. And I said, you've got to keep good records. Otherwise, you bring me something and it's just, oh, here's a process. And I say, how did you develop that? And if you can't tell me accurately and you can't document it, it becomes very problematic for me to accept that process without a lot of input with this thing. So some helpful hints, again, going along the line. FIO testing, we went through that, talked about it. Make sure you understand what it is you're wanting to do with those things. Don't implement full GMPs too early, all right? That can definitely cause you just to bog down, and people will find a way to get around it because they're trying to work. And if it's too locked down, you can't function, people will find a way around it. And I can tell you from doing investigation work for the agency, I can tell you right now, a lot of those things were because people got around the system that was too cumbersome for the process that they were trying to do. And don't forget, you will continue to improve the process even after it goes to market. You've got to have that ability to get that data back and look at it. So, question at the end of this thing, you look at it. Is there a difference between the things we're talking about now, the gene therapy, the cell therapy, tissue therapies that are going on, and good practices that have been pharma for in this industry for years? Yeah, some. I don't want to say not much. There's some technology is there. The approach of the product is there. The knowledge is being gathered, but use some of the things that pharma has been dealing with for years and use that to your advantage, at least as a starting point, baseline, to get a philosophy in this industry of how do you proceed. So let me end it with this, and I'm actually going to hand it off my colleague here, Melissa. Anything that I missed with this?
So there were a couple of questions, and
Okay.
I don't know if Marla is going to jump in.
Yeah, I can ask the questions for you. And so I think the one thing is, as you ended, is there any difference between the GMPs in the GCT world versus what we've been doing in pharma for years? And one of the comments that came through was the speed. So for example, let's see, do we need more controls for GCT as these products move very fast? And then someone else chimed in saying we need faster processes. So do you have any comments around the speed at which we need to move in this world versus traditional pharma that takes 15 years to get a product on the market?
Yeah, that's a great question. I can tell you, yes, you have to be flexible, nimble. You have to have a team and the ability to move quickly on this stuff. The problem with implementation of classic pharma is it was a very rigid system. It's set to do that, to make things go through a very long process to get there. The philosophy is there. So understand why I'm doing this in classic pharma. And it was to control, to ask those questions about what it is we need to do. You've got to be able to take that question and the philosophy into a world now that moves very fast. And a lot of people are out there doing things for rare diseases. So you've got the fast tracking type of things, people that are dying. So it can't go through 15, 20 years of a process. These are things that have to go through very quickly. Again, and it's a very, very good question, is put it into context in this unique organization. If you are there and you have to move through very quickly, you have to have the ability to move fast. What system is it needed to move quickly? That's why I proposed protocols, something that can be written, something that can be documented quickly and put together in a documentation system. It typically moves much faster than a typical process in pharma where you've got a very rigid type of thing that has to go through all these layers of bureaucracy sometimes. Move very quickly with it. I can tell you, and it's very difficult to tell you specifically how to do it, but all these things have to be done, but it has to be done quickly. And you're right. It does have to be done quickly.
Yeah, and I think what people probably don't want to hear is that it's going to require coordinated effort, and that takes time and effort. You have to plan, and you have to get alignment with groups that are in other cross-functional areas, and you have to have meetings that you can then make the decision and move on. You can't just pass a document around that takes weeks and months and finally you get something. So to your point, Troy, if you can set things up in advance, like in this situation, this is our protocol, this is how we go forward, and then the decision part is done and you just do the work.
Yes.
A similar example is in the flu vaccine world. So CSL Behring, I think we have some people joining us today from CSL Behring with their Securus facility in North Carolina where they manufacture the flu vaccine. They don't get from the CDC what they need to work on until like the beginning of the year, and they have to have everything approved and ready to go by the fall. And the way they operate is they have a ton of coordinated action. I mean, it's like you've never seen before. So it's not this easy, okay, here's a process, you just follow it and you can go faster. It really takes a lot of effort to get that alignment at your plant site or, across your entire enterprise, whatever's needed. And that goes along with one of the first questions that came through is, how do you work with R&D to set up qualification parameters for methods and change control for phase one? Because, if you're in quality, for example, down the road, you can't wait until you find out R&D didn't do something that you needed. And it's going to require that there's a high level expectations set by the vice president levels that, yes, we will work together and we'll work together early on. And that's an expectation. If you're just, in regulatory affairs or quality or tech ops or in manufacturing, and you're trying to get the attention of R&D by yourself, it's not going to work. But I'll turn it over to Troy or Melissa to comment on either of those.
I actually think that as far as the speed of the process or the speed of the movement, some of it is it's really how quickly the cells or the gene therapy needs to get from, pulling the cells from the donor or from the patient, processing those cells and back into the patient as well. So we can look at the speed, not only in the development of the process and approval of the process, but in the processing as well. And I agree with Marla, a lot of it is coordination, but documentation of critical processing points, of critical testing points. If we, as Troy brought up during the presentation, if we've already administered the doses to the patient and, then we're doing testing post-administration, what does it look like if we get an OOS? How do we deal with that? So having those procedures and those processes in place, what testing needs to take place prior to administration, what testing needs to take place post-administration. So what does that monitoring look like? So all of that needs to be defined up front and spelled out. Some of it may not be GMP, some of it may be GCP. So it might be good, or I'm not GCP, GTP, good tissue practice. So yeah.
Thank you.
Yeah. And I can tell you, just to add to that, how do you work with people up front, and how do you work with people not even up front and say in R&D, but all across the timeline? It is to make sure that everybody has the vision in their mind of the endpoint. What does it look like? A lot of times R&D will look at things as their job is to make something as good as they can and to toss it over to the next group so they can continue developing it. It has to be coordinated. You've got to have people all along that process line understanding success. So you can't be R&D saying I made something, or this group saying I did something and I'm successful, but the ship sank regardless. We all sink. So have a good vision in place, and then each of the teams has a mission to meet that vision.
Great, thank you. And let me do a time check for Troy and Melissa. Do you have time for me to go through the rest of the questions, or do you want to handle them by email and we get it out to people?
I have time. Certainly I have about, I'd say 10, 15 minutes.
Okay. And for those of you on the line, we will continue to record. So I know we're at the top of the hour. If you need to drop off, you can, but then you can check the recording later to see what the responses were. But with that, I'll just continue then. The next question is, how does the phase-appropriate gradient get applied to the master and working cell banks? They are made in early phase, but used in commercial.
So the master cell bank, again, it's understanding what is it you're trying to control there? Why? What does it mean for you to use that stuff later on? And put it together. It really comes down to, what am I trying to control? Why am I trying to control it? And then control it. So GMPs are all about control. They're the one thing the FDA always looked at. What I always went in to look at was, are you in control? How do you know it? So the master cell bank, the viral vectors, these things all coming together with this stuff. Make sure you put down, what is it that I'm trying to control there? And understand, as these things, yes, come in later, you may have to alter that standing as you progress down the line, but at least you now have it mapped out.
Keep in mind too, these components could be part of a filing. So there are going to be questions, as Troy pointed out. So you want to make sure you have the documentation. what was tested, what was the result of that testing. All of that is going to be questioned at some point if you progress far enough down the path, right?
Yeah.
So if you don't have any of that documentation, it's going to be really hard to prove what you started with and what the result was. So again, starting with the end in mind, all the way back to the beginning, where are we going on this journey? What is our ultimate goal? Because if you get to the end and you're putting together that regulatory filing and you don't have the paperwork from the beginning, then you're lost. So you really want to make sure you've got all your I's dotted and your T's crossed, because you can't recreate it.
Great, thank you. And you may or may not be able to answer this question depending on if you've had direct experience with this or just happen to know, but when is the appropriate time to engage the FDA or regulatory authorities for a company building a new manufacturing facility?
Oh yes, I
think
definitely.
we both can answer this one. Troy, I'll let you chime in, and then if I have stuff to add, I'll jump in there.
So, well, let me say it's kind of a trick question. A lot of times people look at it to say, I'm going to build a new facility, and I'm going to take the design to the FDA, and they're going to approve it. The FDA won't approve it. When is the time to take that sort of philosophy to the FDA? I tell you, if you have a good relationship with the FDA, put together your user requirements. What are you trying to do and why? And then your basic framework of a facility to control those aspects. Then you can go to the agency, talk to them about setting up a quick meeting. Just say, we just want to review it, make sure you're engaged with it. We are here. They are going to say, oh yeah, this is, we approve it and we sign off on it. But you can at least start getting them engaged with it. I have personally taken blueprints and user requirements and where they all fit together in the framework of the facility design to the agency and talk to them about it. And they were very appreciative of being engaged at that early phase.
Great, thanks. Melissa, do you have anything to add?
No, I was going to say the same thing. They may give you feedback or give you suggestions, but they're not going to actually sign off on it, because things change along the way, right? I mean, they're not your quality assurance group or your safety group, or, you're going to have change controls. Things happen along the way. And at some point they may come in and inspect and find differences or find changes that were made that they may not agree with. So yeah, getting some opinions or buy-in or feedback early on, I think, is important. But understand that doesn't mean that once they've given you that, it's forever,
Great. Yeah, thank you. And also, people don't understand that sometimes when you speak with the FDA and they give you their opinion, that the FDA has said. It's like, well, no, that individual has said.
Exactly.
And, you have to make sure that that really is what the FDA, senior leaders would think, because someone else could come into your facility and have a different opinion.
Right.
So
they're just like you and I, right?
Mm-hmm.
The three of us, Troy and Marla and I, all have different opinions.
Mm-hmm.
We're human, just like everyone else. And
right.
Those opinions may change slightly from person to person. So
right. And of course, they're speaking from, understanding the regulations because they are in the FDA. But to your point, it's like they have different levels of experience and opinions. And just recognize that when you're talking with them, are they giving you official response, or are they giving you thoughts and reactions and opinions? So that's something to just be careful of and understand who you're talking to. So another thing, I guess two more points. First of all, some of you put some great resources in the chat box for everyone to access. So thank you for that, whether it was the EU regulations or an ISPE reference document. So thank you. So Troy, you had mentioned that, the technology is a big piece of this and especially the testing. Can you describe a little bit in this world, is it any different than coming up with the testing you need for your pharmaceutical product? Like what's so challenging?
Well, it's like pharma was 30 years ago with new things coming online. think of back, I hate to say it, back when, we were talking about lyophilization of liquids. And what does that mean? How does it work? And putting that together. So technology is being evolved to make those things happen. Same thing now is just, what do you do with this stuff? It's nothing that's really brand new. We're not trying so much to invent a new technology. It's taking some existing technology, making it work, maybe some tweaks to certain things. The technology is there. The more I tell you, the testing is of question. Is like, what do you test with cells and growth of those cells in an environment that you have cell growth going on? And how do you show that's effective? How do you show potency of cells growing? How do you do that? Those are the things you got to come up with. That's really, I tell you, a lot of the questions with it. And when you're talking about editing genes, you're talking about changing things in the human body. How do you show potency? How do you show effectiveness? Think about the vaccines. You brought up a very good point with it. Think about the COVID vaccine. Is it effective? People get the vaccine, but still get the virus. You've got all these things that come into play with it. And it's tough to come back and say, I need to look at these things. And testing sometimes is being developed to show a particular outcome of something that you've made. I'm looking at X. How do I understand X? I test it. And then I find that really didn't reflect the true nature of the product or the process. So you have to go through and then say, what else do I have to look at? What does this mean? Right now, I can say I'm not a QC person, but I've seen enough of it to say I know people are looking at it from that end and understanding, developing the methodology. It's truly the same in classic pharma with a chemical synthesis. What is it you have to look for? Impurities, degradation profiles, all those types of things. It's the same nature now in cell and gene therapy. It's just with a living organism that has to be frozen, typically cryopreserved very quickly within a certain time frame. All those things come into factor with this. So I hope that explains it. I'm trying to explain something very complex in a very short period of time.
Yeah, no, I think that's perfect. Thank you. And this is another question that might be a little bit difficult to answer. I love when people tell me, well, show me where it says FDA says you have to do stability studies of eyedroppers inverted and upright. I'm like, okay, let me find that specific point instead of we know that's the expectation. Yes. So kind of along those lines, it sounds like this person might be wanting some help, like help me prove to my organization that we don't have to go full GMPs so early on. Like, what would you suggest they do to justify, other than, the scientific rationale of, well, we can't lock ourselves in, otherwise we can't make changes. How would you help them, I guess, make that argument?
I tell you, the key thing, and I've met many companies and gone through this very same question. I pull up the FDA's document, the ICH Q7A, and I say, look, I didn't invent this. I was back when we were talking about API GMPs and understood way back in the dark ages when I used to ride into work on my horse. I was thinking about how the GMPs were applied across something like that. So I pull up the FDA's own guidance that they look at, that ICH. The FDA follows that. And I say, look, here this is. I didn't invent it. I'm just taking the same philosophy that they had and saying, I can implement this here as well. And at least plant the seed in their mind that, look, this isn't anything that is new. This has been in place for decades. And we're following something very similar to what they talk about, especially in the context of just the development of it. And again, I'm not talking about no GMPs. I'm talking about specific requirements of control in the various phases of developing this product and getting it to clinical and then getting it to market.
Right. That's a great suggestion, especially since it's a resource that's out there. And you'd mentioned at the beginning as well, ICH certainly is recognized by FDA. So that's fantastic. And to your point, I mean, early on, you need to document your assumptions and decisions and what the outcomes were so you don't repeat failures, right? But so it makes common sense, but you don't have to have like change control in place where everybody's got to sign off. That'd be crazy so early
Correct.
on.
Correct.
Okay, so maybe one last question, and I'll ask Melissa first. You know, if there's a failure that happens in this space, I've heard, you know, I haven't worked directly in this space, but I've heard that sometimes the doctors would look at the patient and say, well, this just didn't work for you, because I mean, it's the patient's blood, it goes back into the patient, and you know, I don't know what else to tell you. This is the process we used, and it didn't work for you. But Melissa, I mean, you might be able to give some insight on how would you know that something that the manufacturer did actually failed? And how would you know who all it could have impacted so you could prevent more issues from occurring with other patients?
So, you know, I don't think this is really that different than classic pharmaceutical. You know, if a failure has occurred or the correction didn't take place, we're going to investigate very similar to a normal investigation. You know, we would go back into the process and see if the processing took place the way it should, the testing took place the way it should, and make sure that all of the aspects of the processing, testing, etc., anything that you would investigate in a normal out of spec investigation or process investigation, you know, were done accordingly and document that. In some cases, you know, these are biological products. So it sometimes it's biology. It doesn't always work for everyone. In some cases, it's a matter of, you know, the science behind it. So, yeah.
Right. Okay. Yeah, that makes sense. I mean, if you're doing something to do a modification and someone realizes that something wasn't done correctly, then you can look back like, well, when was the last time we did it correctly? And then everything else has been impacted. So you can still do your bracketing and do your investigations looking for root cause and impact analysis. So it's got a lot of that same, you know, aspect to it. And then Troy, you had explained to me the other day that there's sometimes when you take the blood product out and that blood product is processed and it can be used for other patients. And so, you know, you can track where it went and things like that. Right. So in many regards, it is similar to what we've been doing in pharma for many, many years.
Yep.
So,
Yep.
Great. Okay, well, I want to thank both of you for taking the time to share your insight and experience in this space that we all know is up and growing and rapidly expanding, but so few of us have actual experience in it. So thank you so much for opening our eyes to what your world looks like and helping the industry to advance.
Excellent. You're very welcome.
All right, and thank you again for sponsoring this webinar so that this is a possibility.
Excellent. You're very welcome again.
All right, well, take care, everyone, and have a wonderful rest of your day.
All right. Thank you.
Thank you.